Hi guys, so it may sound like science fiction to you. Scientists are now able to manipulate the DNA of any living organism of their choices with an incredible efficiency and accuracy. This is achieved by the CRISPR Genome engineering technology, and is definitely going to change both the way research is conducted and the way genetic diseases are treated. So, what's CRISPR? It's short for clustered regularly interspaced short palindromic repeats. Don't panic. CRISPR is basically composed of a set of repeated DNA that acts like an immune system for bacteria and helps them to define viral attacks. Suppose we have a bacterium here, and a virus is infecting it by injecting its genome, that is, the whole set of its genetic information, into the cell. The viral genome is now going to persuade the cell to make lots of viruses and eventually kill the cell. Now, if the bacterium wants to survive, it must stop this replication by destroying the foreign genome. Here is where CRISPR comes into play. Between these repeats mentioned earlier, there are some short sequences called spacers. They match the original sequences in the viral genome, so they act as the bacterium's wanted list, which encode memories of previous invaders. So what if the infecting virus is an unfamiliar face? Then the CRISPR system will employ special proteins to cut out the spatial sequence and integrate it between two repeats. Now the real battle has begun. The whole CRISPR array, including repeats and spacers, are transcribed into CRISPR RNA or cRNA, which encodes the full wanted list. Other CRISPR effector proteins are produced. They are like mobiles piloted by cups and survey inside the bacterium. But if the cups don't have a wanted list or the arrest warrant, they just can't do anything. However, the RNA of full CRISPR array cannot be loaded. Just think about a group of teens riding the same car, quarrel over where to go, and end up going nowhere. In type 2 CRISPR system, transactivating CRISPR RNA or tRNA CRISPR RNA will bind to the repeat, and then the whole CRISPR RNA is preserved into segments containing only one spatial sequence and one repeat, to be loaded onto the molecular vehicle called Cas9 nuclease. Now this complex of Cas9 nuclease, CRISPR RNA and trans-CRISPR RNA, equivalent to a car of cups with a specific mission plans, are about to seek a corresponding sequence in the cell. But here appears another interesting question. How does the complex distinguish the viral sequence from the spatial sequence? The answer relies on a short sequence called protospacer adjacent motif, or PAM in short, which immediately follows the sequence targeted by Cas9 nuclease. It's essential for Cas9's targeting action, so the CRISPR array without PAM will be protected from self-destruction. Once the Cas9 complex binds to the correct viral sequence, two catalytic domains in the protein will act like molecular sealers and cut two strands of DNA separately, introducing a double-strand break and thus destroying the viral genome. Now the bacterium is safe from viral replication, and everything's going on pretty well again. Well, you may wonder how scientists harness this bacterial immune mechanism to make specific modification to genomes, knowing that it can target and cut any wanted sequence. The trick here is that scientists discovered that double-strand breaks in a cell's genome, but not a viral genome, are fixed by DNA repairing machineries, and there are mainly two mechanisms: non-homologous end joining and homologous recombination. In the non-homologous end joining pathway, broken ends are brought together, regardless of any template or similarity, so the fix often causes mutation. Scientists utilize non-homologous end joining to knock out genes of interest, to study the effect of them. In the homologous recombination pathway, a template with sequence similarity is provided and replaces that break, so taking advantage of homologous recombination, scientists can repair mutated genes and even introduce new elements to pre-existing genes. In simple terms, these two mechanisms endow scientists the power to destroy and to create. Now you could see the huge potential of this technology. Scientists are now able to investigate genetic diseases like cystic fibrosis, cure cancer, control malaria by modifying mosquitoes, and even edit human embryos. The future of CRISPR genome engineering is unlimited, and the only hindrance is our imagination. Embrace it, and you might be the one who makes a breakthrough.